Hi BioMonsters, Miss Hines here, and today we're going to be talking about DNA structure. So let's go ahead and get started. So if we're going to talk about the structure of DNA, it's important to ask ourselves, what is DNA? DNA stands for deoxyribonucleic acid. And you're going to want to write down that full name for DNA on your paper. Now, each of the parts of this name of DNA, deoxyribonucleic acid, tells us something about the structure of DNA, but we'll come back to that later. First, let's talk about who discovered DNA. So the discovery that DNA was a double helix was credited to these two guys in this picture. Their names are James Watson and Francis Crick. And this is a picture of them standing with their model of DNA, which again is a double helix. But the truth is that they did not come up with this structure on their own, even though they're given most of the credit for it. In order for James Watson and Francis Crick to figure out that DNA was a double helix, they had to get some help from this lady here. Her name is Rosalind Franklin. Now, Rosalind Franklin was really good at something called X-ray crystallography, which is basically taking X-ray pictures of really small molecules like DNA. This is a picture, and a very famous one, it's called Photo 51, that Rosalind Franklin took of DNA, and it showed definitively that DNA was a double helix. Now, here's the bad part about this story. James Watson and Francis Crick actually took this picture from Rosalind Franklin without her permission and used it to build their model. And did they give her any credit for it? Nope. And so they got the Nobel Prize and Rosalind Franklin got nothing. But today we recognize that she played a really important role too in the discovery that DNA was a double helix. So at this point, you need to go ahead and pause the video and answer the question on your paper. When you're finished, have your teacher check your answer for correctness. Okay, so now we know the basics about how DNA was discovered. We're going to start talking about specifically what is DNA made out of. Now, in Unit 2, when we learned about the chemistry of life, we learned about the 4 groups of macromolecules: proteins, carbohydrates, lipids, and nucleic acids. And DNA's name gives it away which one of those it is. Remember that NA in DNA stands for nucleic acid. So DNA is a nucleic acid. A nucleic acid is a polymer, a really big molecule. And we learned about in Unit 2 that these big molecules tend to be made out of smaller parts called monomers. And the monomer of a nucleic acid is something called a nucleotide. Remember, N with N, nucleic acid, nucleotide. So this is all review from Unit 2, but now we're going to go into some more detail about what nucleotides, the building blocks of DNA, look like. So each nucleotide has 3 parts, and you're going to want to label these 3 parts on your daysheet. The first part is something called a phosphate, and we usually represent that using a circle, and that's what's drawn right here. And so you're going to want to write the word phosphate next to your circle on your paper. The next part looks like a candy house, and remember, candy houses tend to be carbohydrates, and this is actually a sugar. It's a carb. And the name of the specific sugar that is inside of the building blocks of DNA is deoxyribose sugar. So you guessed it, that's where the D in DNA comes from. So the deoxyribo tells us the name of the sugar, and then the nucleic acid tells us that DNA is a nucleic acid. And the very last part of the building blocks of DNA of a nucleotide is something called a base, or a nitrogen base, or sometimes even nitrogenous base. And we'll talk about what that means in just a sec. So make sure you have all 3 parts of the nucleotide labeled on your paper. Now, here's a picture of a nucleotide where it's not drawn using simple shapes, but it's actually showing the full chemical structure. So as you can see, there's lots of different atoms and bonds that make up a nucleotide. But let's just make sure that we can still recognize the three parts even when we're given this really complicated structure. So again, over here, this is the phosphate group, which remember we represented using a circle. And the reason it's called a phosphate group is because it contains the element phosphorus, which is abbreviated with a P. And in the center part, I see this candy house looking structure. So you guessed it, this center part is my sugar, which in DNA is deoxyribose sugar. And then over here, this is the part that I usually use a square rectangle to represent. This is my base, which is sometimes called a nitrogen or nitrogenous base. And as you see looking at the chemical structure, the reason for that is because these bases contain a lot of the element nitrogen, which is abbreviated with an N. So whether you're given this structure using simple shapes or more complicated chemical shapes, you should be able to recognize the three basic parts. Now, we need to go into a little bit more detail about the nitrogen bases inside of DNA. So there's actually 4 different bases or chemicals that can make up that nitrogen base part, and we usually abbreviate them using just their first letter. So the first base is something called adenine. Here's a picture of its chemical structure, and we're going to abbreviate adenine simply using the letter A. So we say that there are A's inside of DNA. Another base that we find inside of DNA is the nitrogenous base thymine. Here's a picture of its chemical structure, and we're going to abbreviate it using the letter T. Next, we have a cytosine, and here's a picture of cytosine's chemical structure, and we're going to abbreviate that just using a C. And then lastly, of course, we have guanine, which we're going to abbreviate using a G. And over here is a picture of its chemical structure. So write down these abbreviations for the 4 nitrogenous bases on your paper. Okay, at this point you need to pause the video and answer the stop and jot question on your paper. When you're finished, have your teacher check your answer for correctness. Okay, now that we know the basic building blocks of DNA are nucleotides, we're going to talk about how do we put these building blocks together to actually form DNA. So in order to form a single strand of DNA, we have to connect multiple nucleotides. And the way we do that is that the sugar from one nucleotide connects to the phosphate on another nucleotide. So I'm actually going to draw this connection in my picture, and you should do the same on your paper. So if I wanted to connect these 2 nucleotides together to form a piece of DNA, I need to connect the sugar on one to the phosphate on another, and that's my bond right there. And I could do this again and again and again, thousands of times over, to form a very long strand of DNA. Now, that's not the whole picture, because we know that DNA is a double helix. It actually has 2 strands or 2 sides. So on the sides, my sugars are connected to my phosphates. But how do the 2 strands or 2 sides of DNA connect to one another? Well, the 2 strands are held together by hydrogen bonds between the bases on either side. So this base on this side of the DNA will form a hydrogen bond. So I'm going to draw a dotted line to represent that bond to this base on the other side of DNA. And this base will do the same thing. And that's how my 2 sides of DNA are going to connect to one another. So you should draw those connections on your paper as well. And when we put it all together, what happens is that the DNA starts to coil up and form this helical shape that we've come to know and love. Okay, at this point you need to pause the video and answer the stop and jot question on your paper. When you're finished, have your teacher check your answer for correctness. So the next thing we need to start talking about is how the bases pair up with one another, because it turns out that they follow a specific rule for which ones will hydrogen bond to which ones. And this rule was first discovered by this guy, a scientist named Erwin Chargaff. And what he did is he took a bunch of DNA out of different organisms. He took DNA from an ox, from some human cells, even from an E. coli, little bacteria or prokaryote. And he analyzed the composition, or what was inside of that DNA. And what he noticed is that no matter if you were an ox or a human or a bacteria, your DNA always followed a certain rule, which came to be known as Chargaff's rule. And that rule says that the amount of adenine, or A, in your DNA is always equal to the amount of thymine, or T, in your DNA. And the amount of G's, or guanines, in your DNA is always equal to the amount of cytosines in your DNA. So Watson and Crick took this rule and formed it into what has become known as the base pair rule. The base pair rule states the following. It states that A, or adenine, will only bond, or hydrogen bond, with thymine, and that C will only hydrogen bond with guanine. And if you look at this picture, you can understand why. A and T will only fit together with one another. If you tried to put an A with a G, it just simply wouldn't work. And if you tried to put a G with a T, it just wouldn't work. Only A and T can bond with one another, and C and G. So we actually have a few different mnemonic devices to help you remember the base pair rule, and you don't have to write all of these down on your paper. You should pick the one that works for you. So one mnemonic device to help you remember this is apple trees and collard greens. Apple trees has an A and a T in it because A and T will only bond or pair with one another, and collard green starts with a C and a G because C and G will only pair with one another. So apple trees and collard greens is our first mnemonic device. Or if you prefer, you could say apple trees and go-karts, same thing, A and T and C and G. Or you can simply remember at GoCo. A and T for the at, because A and T will only pair with one another, and then GoCo, C and G. At GoCo. So you should choose whichever one of these mnemonic devices you think will help you remember the base pair rule and write it down on your paper. Now, the base pair rule has some significant implications for the structure of DNA. What it means is that if we know what bases are on one side of the DNA, we can predict what bases are on the complementary or opposite side of DNA. So for example, if I know that the nucleotides on this side of DNA contain the base A and the base C, I can predict what nucleotides must be on this side of the DNA. This nucleotide must contain the nitrogenous base T, because A only goes with T at GoCo, and this one must be a G, because C only goes with G. So again, if you're given one side of the DNA, you should always be able to say what letters or bases are on the other side, or complementary side. Okay, at this point, you can pause the video and answer your stop and jot questions. When you're finished, have your teacher check your answer for correctness. So the last thing we want to mention today is just why do we care so much about DNA? Scientists are always talking about DNA, and why is that? Why are we so obsessed with this molecule? And we're going to be talking a lot more about this in the coming weeks, but we want to just give you a taste right now. So the reason we care so much about DNA is because DNA stores our hereditary information. But what does that mean? It means it has something to do with our genes, but again, I don't really get it. Why is that so important? Well, really, what that means is it means that the sequence of nucleotides, so our building blocks in DNA, especially the sequence of nitrogen bases, which remember is the letters A, T, C, and G inside of DNA, is the code for how to make proteins. Ah, interesting. A different macromolecule group. And the reason we care about proteins is because it's proteins that determine our physical traits. So here's a picture at the bottom that illustrates that. So this is a segment or a piece of DNA. And maybe your DNA has the sequence of nitrogen bases A, T, G, G. And this sequence might code, it might tell your cell to make a particular protein. And it's this protein that maybe makes your eye color brown. Whereas somebody else would have a different sequence of nucleotides or nitrogen bases that would give them a different protein that would give them a different eye color. So really, the DNA is going to determine your physical traits through what proteins it codes for, and that's why we care so much about it, and we're going to talk a lot more about that. So go ahead and pause the video and answer your last Stop and Jot question on your paper. When you're finished, have your teacher check your answer for correctness. Thanks.